Hello class, I would like to walk you guys through the cell membrane and transport notes. Let's start here with the gummy bear mass changes. We have the average change in mass over here, how much the mass was gained or lost, and we have the salt concentration over here. So here we have no salt, and over here we have very salty, 1 to 2, 1 part of salt to 2 parts of water. And here we have 1 part of salt to 5 parts of water, so not quite as salty. And as you can see, in just the normal water, the mass changed the most. And over here, in very salty, it actually lost mass. So what happened? What made that change in mass occur? Why did the gummy bears get bigger or smaller? So what we're trying to do is to apply this information to what is happening in the cell. So to understand that, we need to know the cell membrane. And of course, we build those membrane sections before with the folded paper. And really what we're looking at here is just a section of the larger membrane that goes all the way around the cell. So the plasma membrane is our boundary between the cell and the outside environment, and it's going to help regulate all the transport that happens from the cell. If anything needs to go in or out of the cell, it's got to go through the membrane. So if we want stuff in like food or oxygen, it needs to go through the membrane. If we want stuff out like waste or proteins that have been made, we need to go again through the membrane. So really what you're going to want to draw is this kind of diagram right here. It's really basic. You've got the phospholipids. The individual lipids are just a circle with fatty acid tails. You've got a bilayer of them, meaning you have a row of lipids here on top and a row of lipids upside down in the bottom. This is to help keep the water out because these fatty acid tails here, they don't like water, and so they orient themselves away from it. These guys can handle water. They don't mind it so much. So if they're just kind of floating around in water, it's not really going to be able to get passed through unless there's a change in concentration and a change in pressure that would make the water want to move. And of course, we have these proteins here. So the whole thing is the membrane. It's a mixture of lipids and proteins. And we can assume that outside the cell could be here and inside the cell it could be here. So it could be reversed. We don't really know in reference to this part, but it's definitely not within this part. That's definitely not inside the cell. So again, what's happening with the gummy bear? Well, it's a type of diffusion. Diffusion is really just molecules moving around at random, and they're going to follow a general pattern of high concentration to low concentration. So these little circles are bouncing around all the time. And usually they're going to find their way to where there's a less crowded place. So with all this crowded room over here, we might expect some of these circles to travel over to the other side since the triangles can't really move at all. So when you need to draw a concentration gradient, this is really what you're looking for here. I've got high concentration and then a low concentration. And high concentration of whatever molecules I'm talking about. So they can pass through a membrane, and we would call that passive transport. This passive transport is stuff moving in and out of the cell with really no energy needed as that happens. It can just move in and out of the cell because they're small or because there's just a general change in pressure of things being forced one way or another. So when this happens specifically with water, we call that osmosis. So osmosis is the diffusion of water. It's water moving in and out of cells, and it depends on whether there's more or less water somewhere, but really that's relative. Relative to what? Well, it's relative to the solutes. The solutes are the salts, the sugars, the things mixed in the water. So over here, I have a lot more solutes than over here. So if I was to leave this alone over time, since there's actually less relative water over here, water is going to move in that direction, high to low concentration of water. But that's actually going to mean a low to high concentration of solutes. So what does that mean for our cell? Well, it's like what it meant for our gummy bear. When the gummy bear was in very fresh, not salty water, it grew. Water went into that cell, into that gummy bear, because there wasn't as much water in the gummy bear or in the cell. So this could increase the mass. If it's a plant cell, it could swell up. If it's an animal cell, without that cell wall, it could actually rupture, which is called lysing. This cell here has lysed, but the movement of water has gone into the cell. So these are really what we're drawing for those different solutions. So the solution is the concentration of water in the environment around the cell. So if it's hypotonic, there's actually more water on the outside and less water on the inside. If it's isotonic, there's an equal amount of water relative to the solutes inside and outside. This is what we're going to look for if we're going to try to put water in someone's veins with an IV. It's going to be kind of salty water. But if we gave somebody very salty water, then their blood cells could even shrink because all the water is getting pulled out of them by that salt. So this is called a hypertonic solution. Iso means equal. Hypo means a lot, as in a lot of water. Hyper could also mean a lot in terms of lots of salt. So iso is equal, hypo, hyper. Hyper is generally a lot more salt, a lot less water. Hypo, a lot more water, but a lot less salt. And we could replace the word salt in all these situations with sugar. Now, let's say I want to move a lot of stuff at once. That's where proteins come in handy. So these protein channels can help things move in or out of the cell that are maybe too big to go through the membrane, or maybe I just want to move way more that can fit through the membrane at once. So it's still going to follow the concentration gradient, this high to low movement, kind of like moving downhill. So there's really no energy required by the cell. It doesn't have to do anything. This can just happen on its own. It just has these proteins around to help it out. Now, this is passive transport, but it's still diffusion, but it's being helped. So we'd call it facilitated diffusion. Diffusion that's being facilitated is to be helped along. Now, I have a comic here. We have 2 men in a jail cell, and he says, "What you got there, Jim?" And he says, "Transport proteins." And as you can see, he finds his way outside of the cell, kind of like what we saw here. So hilarious. And finally, let's say I don't want all this water in my cell. Let's say I'm a swelling gummy bear, I'm a swelling cell, and I have to get this water out. Now, it's moved into my cell because of the concentration. So now I got to get it out of the cell, which is going against the concentration. Or maybe I need to force waste out of my cell. I can't just wait for it to happen. So I need to use energy. And how do I do that? Well, we've got these other transport proteins that are activated by energy. And the energy of the cell is called ATP. It's called adenosine triphosphate. And it's what's made from the mitochondria. So when we say it's the powerhouse of the cell, it's producing the power. It's producing the ATP from respiration. So this ATP can activate our transport proteins and help force things in or out of the cell against the gradient. So if there's a lot of a certain molecule outside of the cell, but I still want to get my stuff outside of the cell from low to high, I got to force it that direction. So it's like forcing it uphill as opposed to downhill. But I need energy to do that. So in summary, we've got 3 main types of transport for things moving in and out of the cell. We've got simple diffusion, which is just from high to low concentration. It can happen across the membrane without any help at all, just whether there's high or low concentration. Now, facilitated diffusion is very similar, except maybe they can't fit through the membrane, so they need some help. They got to go through a protein. Both of these are passive transport because they don't need any energy to do so. They just follow high and low concentration. Now, if I want to go against the concentration, I want to push it or pull it to where there's already a lot of them. So let's say it's sugar here, and I really want more sugar in my cell, so I got to pull it in, even though I already have a lot. I might need energy to do that. I need ATP as the energy to do that. All right, that's cell transport and cell membranes, and I hope that was helpful.